Today's lecture will delve into the cardiovascular system as we look at blood vessels and the cells within the blood. The circulatory system is composed of muscles and networks of tubes that are able to pump cellular components of our blood to all the tissues in our body. I think it's an amazing statistic to think that we have between 100,000 to 150 kilometers of blood vessels in our body. That's like driving to Florida and back 24 times. Just incredible. At the center of our cardiovascular system is, of course, the heart. The heart will pump blood into the arteries, so that's the efferent vessels, so vessels carrying things away from the heart. The arteries will become increasingly smaller as we move away from the heart until we hit the capillaries. The capillaries represent the smallest vessels and the exchange site of oxygen between the blood and the tissues. Every organ in our body will have these fine microvascular tubules for exchange of oxygen. The veins are then created when venules leaving the capillaries merge together and give rise to larger veins. These veins will continually enlarge until they finally reach the heart, where blood is returned back into the heart, ready for recirculation. Let's first take a closer look at the heart. So here we have figure 11-2, a schematic diagram of the heart depicting its 4 chambers. So blood, of course, enters into the heart into the right atrium from the veins. It then moves from the right atrium into the right ventricle, which then pumps blood out to the lungs, and then blood returns back to the left atrium and then left ventricle, which then propels blood out into the arteries to the rest of the body. The heart is essentially composed of a very muscular wall with various valves. Now the layers of the heart wall is composed of 3 layers. We have the internal endocardium. So this is composed of a layer of endothelial cells. Recall, endothelial cells are simple squamous epithelial cells. And these cells are overlying connective tissue that contains some smooth muscle fibers, as well as branches of the heart's impulse conducting system. This conduction system is said to exist within the subendocardial layer. Deep to the endocardium is the myocardium. So this is where the cardiac muscle, which performs the pumping of the heart, resides. And then on the outer aspect of the wall is the epicardium. So this is composed of a mesothelium layer, again, simple squamous epithelial cells, overlying connective tissue containing both blood vessels as well as nerves. The epicardium will also fuse and form part of the pericardium. The valves of the heart are composed of dense connective tissue lined on both sides by endothelium. Starting first with the endocardium, here we have the microscopic appearance of the endocardium, figure 11-3. On the outer surface, again, we have our endothelial cells, which we can't appreciate in this image because it's low power. Under the endothelial cells will be some loose connective tissue. Deeper to this is our subendochondrial layer. Within this layer, we're going to see some smooth muscle, and we'll also see the nerve conducting system. This system is actually specialized cardiac muscle fibers that are going to set up conduction instead of contraction in this area. Deep to the endocardium is the myocardium. So in the inferior aspect of this image, we see the deep eosinophilic or pink color of the cardiac muscle. The myocardium represents the thickest layer of the heart wall. It consists primarily of cardiac muscle. You will recall that cardiac muscle, similar to skeletal muscle, contains myofibrils all in an organized layer, giving rise to a striated appearance. Now, contrary to skeletal muscle, the cardiac muscle tend to only have one nucleus, sometimes two or three, but typically one, and it's centrally located. These cells are also branching and joined together via intercalated discs. This architecture allows the cardiac muscle to form a sheet-like configuration such that upon contraction, the lumen of the chamber of the heart will decrease, which in turn will propel blood out of the chamber of the heart. Figure 10-16 from your text is a nice cartoon example of some of the cell-to-cell junctions that we find within cardiac muscle. So within the intercalated discs, we have an abundant number of desmosomes and some other adherent junctions that create this firm attachment between cardiac cells. And then we also have longitudinally along the cells a number of gap junctions which allow communication between the cells. The outermost layer of the heart wall is the epicardium. So this is composed of mesothelium, so simple squamous epithelial cells, and they're overlying some loose connective tissue containing blood vessels and nerves. The epicardium in turn becomes part of the pericardium. Between the epicardium and the pericardium is the pericardial cavity. Within this cavity, we have a small amount of fluid. So the adipose tissue of the epicardium combined with the fluid within the pericardial cavity help prevent any frictional damage to the heart wall. Figure 11-4 from your text nicely depicts the microscopic appearance of the epicardium with its simplistic mesothelium, so simple squamous epithelial cells. Sometimes the mesothelium may look more cuboidal epithelial cells, and this is overlying loose connective tissue containing nerves. Here in this image we have a nice branch of a large nerve fiber. And then deep to this is, of course, the myocardium, making up the middle layer of the heart wall. And recalling from some of your gross anatomy, the coronary vessels and the coronary circulation of the heart, these coronary vessels are located within the epicardium. Also important to the structure of the heart are dense fibrous connective tissue that forms part of the interventricular septi. It also surrounds all the valves of the heart and supports the heart valves as well as the chorda tendineae. This cardiac skeleton provides insertion points for the cardiac muscle, and it also acts as an insulator between electric signals between the atria and the ventricles, so that the correct signals are going to the correct portion of the heart. Figure 11-5 from your textbook of a valve in the heart shows the connective tissue skeleton making up the valve, an extension of the connective tissue into the chorda tendineae, and this connective tissue being covered by an endothelium layer. Deep to this valve is the myocardium. And just a quick reminder that the conduction system of the heart is composed of two nodes, so the sinoatrial node, the atrioventricular node, as well as the atrioventricular bundle, or bundle of His. and they all communicate via the subendochondral conduction network of myofibers termed the Purkinje fibers. And recall again that the Purkinje fibers are located within the endocardium in the subendocardial layer. The heart is under the control of the parasympathetic and sympathetic nervous system. Nerve cell ganglions are located near the sinoatrial and atrioventricular nodes. They will affect heart rate and rhythm. The parasympathetic system functions via the vagus nerve to slow the heartbeat, whereas the sympathetic nervous system accelerates activity of the pacemaker or the sinoatrial node. Free sensory nerve endings are also located in the myocardium. These are fibers that will register pain in the heart. These fibers will, for example, transmit pain signals if someone is experiencing angina. Angina is when someone has inadequate oxygen supply to the myocytes of the heart. Leaving the heart now, we're going to make our way from the left ventricle now into the aorta. So this is the first artery in which oxygenated blood leaves the heart. As the blood moves through the cardiovascular system, first in the aorta, it's starting in a very elastic artery. From the elastic arteries, we move into muscular arteries, then into smaller arteries, into then arterioles, and lastly ending up in the capillaries. From the capillaries, we move into venules, which are the smallest veins. From the venules, we move into small veins, then to medium veins, and lastly into large veins as we return to the heart. So let's make our way through the microscopic appearance of these various types of blood vessels. All of our blood vessels, with the exception of the capillaries, contain endothelium as well as smooth muscle and connective tissue within their walls. The capillaries are solely lined by endothelium, so we have no connective tissue within their walls. Blood vessels, whether arteries or veins, have a similar general architecture to their walls. They're composed of three concentric layers: the intima, the media, and the adventitia. In the intima layer, we have endothelium and its associated loose connective tissue. And if we're in an artery, we will have a certain degree of elastic tissue making up the internal elastic lamina. The second layer, the tunica media, or middle layer, is composed of concentric layers of smooth muscle fibers. There will be a varying degree of elastic fibers existing between the muscle cells. Reticulin fibers may also be noted. In the arteries, there may also be a defined external elastic lamina within the tunica media. And then lastly, we come to the adventitia. So this is the outermost layer, or the tunica externa. And this is connective tissue primarily composed of type 1 collagen as well as elastic fibers. The adventitia is bound to the stroma of the organ through which the vessel is running. This helps keep the vessel in a particular position in space within a given organ. If the vessel in question is large enough, there may be a special network of vessels supplying the wall of the vessel that's termed the vasa vasorum. Now recall from your lecture on epithelium that the endothelium is composed of simple squamous epithelial cells. And the H&E slide in the lower right hand side of the screen nicely depicts a fairly small blood vessel lined by endothelial cells with a little bit of smooth muscle within its wall. Although endothelial cells are very simple in their architecture, they're quite important. They act as a semi-permeable membrane between the blood and the interstitial fluid. They allow for selected molecules to transverse into the tissues from the blood. They function as an antithrombogenic surface. This means that the endothelial cells take great care in discouraging the blood from clotting on their surfaces. They even secrete heparin and tissue plasminogen activator to prevent clot formation. They also play a role in vascular tone and blood flow. They're able to stimulate smooth muscle contraction and thus increase blood pressure. But they're also able to stimulate relaxation of the muscle and relaxation of the vessel via secretion of nitric oxide and prostacyclin. Endothelial cells play a critical role in an immune response during inflammation. They are the major player in recruitment of neutrophils to a site of insult. They also produce hormones. Endothelial cells can produce vascular endothelial growth factor, which is critical for the maintenance and new growth of blood vessels. And they may also release angiopoietin, which will recruit smooth muscle cells and fibroblasts to help form the other tissues that make up blood vessels. So keeping in mind this general structure of blood vessels, so intima, media, and adventitia, let's look at the various types of blood vessels as blood courses through our body. Let's begin first with the elastic arteries. So these include the aorta and the larger pulmonary branches. Elastic arteries primarily function to carry blood to smaller arteries. Architecturally, they have an extremely well-developed tunica media with extensive elastic component. Here we have a microscopic, fairly low power image of the aorta, figure 11-7 in your textbook. And the three layers of the artery is nicely displayed, starting first with the tunica intima. And again, if it was higher power, we'd appreciate a very thin endothelial layer. And then we have loose connective tissue. In the center layer, the middle layer, the tunica media, you can see a great number of elastic fibers interspersed between the smooth muscle fibers. And this particular vessel has both an internal as well as an external elastic lamina within the tunica media. And then lastly, in the tunica adventitia, we do have a fairly developed vasa vasorum, so network of blood vessels providing nutrients to the wall of the aorta. The high elastic nature of these arteries is very critical for the arteries to be able to maintain a uniform blood flow leaving the heart. So not only are they able to withstand the forceful flow of blood after ventricular contraction, but they also can tolerate the sudden decrease in blood flow when the ventricles then relax. The elastic fibers allow the vessel to rebound fairly passively. This therefore helps maintain a fairly consistent arterial pressure. The elastic arteries are also an area where we will find specialized arterial sensory structures. First off, we have the carotid sinuses. These are located at the common carotid artery branch. They act as baroreceptors in the carotid sinus to help maintain arterial pressure by detecting the amount of stretch that the artery is undergoing. This area is supplied by cranial nerve 9 or the glossopharyngeal nerve. We have a very similar baroreceptor that's located in the aorta, and it is supplied by the vagus nerve. We also have specialized chemoreceptors that are located within the same regions, called the carotid bodies and aortic bodies, and these help monitor both carbon dioxide and oxygen levels. Let's move now from the elastic arteries down to a slightly smaller artery, being the muscular arteries. Similar to the elastic arteries, they play a role in distributing blood to organs, and they do help regulate blood pressure through relaxation and contraction of the smooth muscle within the tunica media. Here we have a microscopic image of a muscular artery, this one being in particular the splenic artery. And you can see again the tunica intima with its fine endothelial layer. Remember, at this power we can't really appreciate the endothelial cells. Its tunica media is still fairly prominent but much less developed than the aorta, so it has roughly 40 layers. And it will have still an internal elastic lamina and external elastic lamina, denoting the edge of the tunica media. And then we have the tunica adventitia again, and we can see a vasa vasorum network of blood vessels as well. As the arteries decrease in size, the quantity of elastic fibers as well as smooth muscle fibers will gradually decrease. Here we have a nice microscopic image of a small artery. So you can see very easily this artery is in cross section, and you can appreciate how small the tunica media is. There is no evidence of a vasa vasorum within its much thinner adventitial layer. So this vessel is going to depend on its nutrients and oxygen from diffusion of the blood through the vessel wall. The arteries will continue to branch and get smaller and smaller, and eventually they reach the point where they are composed of only 2 layers of smooth muscle within their tunica media, at which point they are coined arterioles. Arterioles represent the beginning of the microvasculature of an organ. They are generally less than 0.1 millimeters in diameter, with their lumen being almost as big as the thickness of their wall. The subendothelial layer within the intima layer is very thin. There is no elastic lamina within the tunica media, and the adventitia is so thin that it is almost inconspicuous. Arterioles will virtually all arterioles will eventually branch to give rise to an astomosing network of capillaries. This microscopic image at very high power of arterioles is a great comparison for size. The right-hand side of the screen in particular demonstrates the artery, where the lumen of the artery is almost the same size as the thickness of the wall. You can see the endothelial cells lining the intima. You can see 1 or 2 layers of smooth muscle cells within the tunica media, and really can barely distinguish that there's an adventitia around the periphery. The arterioles in this image are of slightly different size. So as the arterioles get smaller and smaller, as they transform themselves into the capillaries, they begin to lose their muscle, their tunica media, and then giving rise to the capillary. So you can see on the upper right-hand side of the screen, denoted by the C, the appearance of a capillary. This is essentially large enough to contain one red blood cell. Interestingly, the smooth muscle fibers that line these small arterioles act as sphincters that periodically close off the arterioles and deliver periodic blood flow into the capillaries. In this way, they are actually resisting blood flow at all times. These sphincters are actually the major determinant of systemic blood pressure. Blood pressure depends on cardiac output and the total peripheral resistance to blood flow. It is the arterioles that are actually creating this resistance. Hypertension is commonly due to an increase in peripheral resistance. Here we have a schematic diagram of a contracted arteriole that has shut off any blood passing into the capillary bed. Moving now to the capillaries. The capillaries are composed of a single layer of endothelial cells, essentially rolled up as a tube, surrounded by their basement membrane. There are no other wall components or layers. They may sometimes have a cell termed a pericyte, and this is a cell that does have some contractive capabilities in their walls. Capillaries represent roughly 90% of our vasculature. Because of the sphincter effect from the arterioles, there is only about 5% of blood volume in the capillaries at any given time. Due to this small volume, and thus low flow, it maximizes the exchange of water and other solutes between the blood and the tissue. And bringing back that previous microscopic image of arterioles again, we can see comparatively the capillary with its endothelial, we can see 2 nuclei composing its wall and its lumen being big enough for roughly 1 red blood cell. There are 3 different types of capillaries. This includes continuous capillaries, which are the most numerous, fenestrated capillaries, and lastly, sinusoids. Continuous capillaries are located within muscle, connective tissue, our exocrine glands, as well as nervous tissue. Fenestrated capillaries are located within endocrine organs. They're also found in the intestinal walls, the kidney, and the coronoid plexus. Sinusoids represent discontinuous capillaries, and these are found in the liver and spleen, as well as the bone marrow, and also some endocrine organs. If we look at the ultrastructure of continuous capillaries, again, we can see this is in cross-section. Looking a little closer at the continuous capillary, so again, the most numerous capillary, we can see that it's composed simply of a layer of endothelial cells. So there's 2 endothelial cells seen in cross-section here, sitting on a basement membrane. Between the endothelial cells, we have very tight occluding junctions. There is a continuous basement membrane wrapping around the endothelial cells. Comparatively, the fenestrated capillary still maintains tight junctions between the endothelial cells, but it contains many perforations or fenestrations within the endothelial cell that allows for more material to pass through the cell. The fenestrated capillary still has a continuous basement membrane. And then lastly, we have the sinusoid capillaries. These capillaries contain large fenestrations with a discontinuous basement membrane around the endothelial cells, and this allows for a maximum exchange of fairly large molecules to occur. Up to this point, we've already discussed the simple pathway that blood will take from the heart into the arteries and arterioles, through the capillaries, and then out of the capillaries into the venules and veins back to the heart. So this is basically considered the simple pathway. Now there are some alternative pathways that may occur within the microvasculature. These include arteriovenous shunt. So this is when the blood enters a separate vessel and shunts clear past the capillary. We also have a venous portal system. So this is when the blood enters into one capillary bed, exits via a vein, and enters into another capillary bed. And then lastly, we have an arterial portal system, which is not displayed in this diagram. So let's look at each of these alternative pathways a little closer. The arteriovenous shunt is quite important. It's found in the skin, so in our fingertips, our nose, even our lips. And this is when the arterioles in the skin will contract to direct the blood away from the capillary bed and to shunt it into the exiting venule. This is very important for thermoregulation. High capillary blood flow will allow for skin to dissipate a fair amount of heat, while a reduction in blood flow through the capillaries will allow for preservation of heat. So the effects of severe frostbite is a great example of the effect of the arteriovenous shunt. So you're exposed to the elements, your body is extremely cold, and in an effort to preserve heat, it's going to shunt the blood away from the capillaries within the hands, and thus the skin will be deprived of enough oxygen exchange and begin to undergo necrosis. The venous portal system is found primarily in the liver, and also a similar example is seen in the anterior pituitary gland. This is when blood flows through two successive capillary beds that are separated by a portal vein. This allows for high concentration of solutes to be delivered to the next capillary bed for exchange without being diluted in the rest of the circulation. And then the last alternative route is the arterial portal system, which we find in the kidney. So this is when the kidney will have an arteriole that is both afferent and efferent vessel, and the blood will flow into the kidney from the efferent vessel through the glomeruli, which represents the capillaries of the kidney, and then will exit the capillaries of the kidney via the efferent arteriole. So the blood is essentially passing through a capillary bed without entering into a vein. The importance of our microvascular circulation is nicely demonstrated by diabetic microangiopathy. Diabetics unfortunately get an increase in the basement membrane thickness of the capillaries secondary to chronic hyperglycemia. This results in decreased metabolic exchange in the capillary beds. The most prevalent affected organs include the kidneys, the retina of the eye, skeletal muscle, and skin. Lack of nutrient exchange in the microvasculature of the skin results in non-healing ulcerations. Similarly, diabetic patients develop nephropathy secondary to thickening of the basement membrane within the glomeruli, which leads to damage of the glomerulus and associated tubules. This is a nice microscopic image displaying the glomeruli of a healthy kidney on the left-hand side with a diabetic kidney that is completely devoid of proper capillary spaces. No red blood cells are able to pass through this completely destroyed glomerulus. The unfortunate damage to the blood vessels supplying the retina of the eye leads to the eye being stimulated to produce new blood vessels. These new blood vessels are produced rapidly and they are very leaky, and we see fluid being leaked into the retina of the eye and causing damage to the eye. Let's leave now the arteries and arterioles and capillaries and now make our way through the venous system. Venules immediately beyond the capillaries are termed postcapillary venules. These particular specialized venules are quite similar to capillaries in that they do not contain a tunica media within their walls. They may have pericytes quite similar to the capillaries. The significance of the postcapillary venules is that they are the site where white blood cells typically leave the circulation when they are being recruited to a site of infection or tissue damage. Moving on now from the postcapillary venules, the blood then drains into the collecting venules. The collecting venules indeed do contain a tunica media layer with roughly 2 to 3 layers of smooth muscle. The characteristic feature of a venule is that its lumen is quite large relative to the thinness of its walls. Figure 11-21 in your text demonstrates a comparison between arterioles with postcapillary venules in the top part of the screen. So you will remark on the large lumen size of the postcapillary venule relative to the arterioles, and also the very simplistic wall, so simply one layer of endothelial cells with occasional pericytes. Now, pericytes are not readily identified in comparison to endothelial cells within a postcapillary venule, and I will not ask you to be able to make that distinction. The lower portion of the photo is depicting the larger collecting venules, and again you can appreciate their very simplistic wall with only occasional smooth muscle cells within their tunica media. In fact, the smooth muscle cells can be often quite difficult to appreciate. It's really the marked lumen size, again, that sets these venules apart from the arterial system. If we were to look at an artery as large as this particular venule depicted in the picture, it would have quite a prominent and obvious tunica media. Figure 11-21C from your textbook nicely demonstrates a venule that it's a little easier to appreciate the various layers within its cell wall, with a more obvious endothelial cell layer, some smooth muscle cells being discretely seen around the periphery, as well as an adventitia surrounding the outside layer. And a very small portion of a larger arteriole is in this image as well. So you can see how much thicker the tunica media is in the arteriole relative to the venule. Figure 11-221D actually demonstrates the significance of the postcapillary venules, again being the major site of white blood cell recruitment. You'll recall at some point learning in physiology how the endothelial cells play a fairly significant role in the inflammatory processes. They aid in recruitment of neutrophils. So you can see here highlighted by the arrows, we have passing through the wall of the postcapillary venules are neutrophils. These are fairly small, quite pink eosinophilic cells with bilobed or multi-lobed nuclei. And then also, just for interest's sake, within the lumen, we can see that classic look of red blood cells. As the collecting venules begin to merge together, they will eventually give rise to veins. Smaller veins will then fuse together and become larger and more muscular, and their walls will be thicker and take on more complexity in that they acquire more elastic tissue, quite comparable to arteries in that regard. Now, an important feature of large and medium-sized veins is the presence of valves. So this is one feature quite unique to the venous system. This schematic diagram is a nice comparison of the architecture of a fairly typical vein in comparison to a typical artery. So you can see, one, the endothelial layers are absolutely identical, but where the difference starts to arise is, one, the quantity of elastic tissue, or even having the presence of a real defined elastic lamina. And then we have the thickness of the tunica media, where the relative thickness of the artery is much thicker than veins. And then we have the adventitia layer, with the vein actually having a slightly thicker adventitia. But when we look at that microscopically, it can sometimes be difficult to appreciate, as the adventitia definitely fuses with the surrounding connective tissue. Now, the structure of smaller and sort of small to medium-sized veins lacking a very thick tunica media makes sense in that these veins tend to be under extremely low pressure. So a real thickened muscular wall is really not essential. They actually are quite under the influence of compression from surrounding skeletal muscle. These small and medium veins tend to run parallel to their corresponding arteries. And again, the smaller veins will not contain valves, but as the veins become somewhat larger and more into the medium realm, they will contain valves. Figure 11-22A demonstrates a small vein with its relatively large lumen compared to a small muscular artery with quite a thick media and defined adventitia, and noticeably smaller lumen in comparison to the vein. Figure 11-22C from your textbook is demonstrating a medium-sized vein and comparing it to a small muscular artery. The particular stain used here will highlight the fibrous tissue as being fairly beige in color. The muscular tissue is quite pinkish to almost purple, and then the elastic tissue is being stained here a black color. So you can see in the muscular artery, we can see both an internal as well as external elastic lamina. And that's the real presence of increasing elastic tissue is what is really defining the muscular artery. Now, in comparison, our medium-sized vein has a very large lumen. We can see a slightly thickened tunica media, nothing in comparison to the muscular artery, but a more thickened and well-developed adventitia. I can appreciate that it's difficult sometimes to distinguish, for example, a small vein from a medium vein. I'm really not expecting you to be able to do that. I do expect, though, in a diagram such as this, that you would be able to distinguish which one was the vein and which one was the artery in this given case. The large veins in the body constitute those vessels which lie close to the elastic arteries close to the heart. The large veins have a well-developed intima layer, a fairly thin media, but still larger than our small to medium veins, quite a thick adventitia. Sometimes some smooth muscle will even spread out into the adventitia. And the large veins will have elastic fibers noted within it, both within the media as well as the adventitia. Occasionally in a given large vein, we may see a defined internal elastic lamina. And recall again, large veins will contain valves. Returning again to figure 11-7 of the aorta, which we saw earlier in the lecture, we now can compare it to the vena cava. So this is obviously the large vein that's returning blood to the heart. And this diagram demonstrates a very good comparison between the tunica media of the vena cava compared to the aorta. Very comparable tunica intimas between the two, even a defined internal elastic lamina within the vena cava, and then quite a thickened, well-defined tunica media in comparison to the tunica media of the aorta, and we'll even see some elastic fibers sitting within the tunica adventitia of the vena cava. As I've already mentioned, valves are located within the medium and large veins. Are we always going to be able to appreciate the presence of valves microscopically? Absolutely not. So every once in a while in these larger veins, we're going to have the luxury of being able to find a valve. The valves are composed of folds of the tunica intima, and they will have an increased quantity of elastic fibers. A higher quantity of valves are located within our legs in order to prevent backflow of blood that's being directed back to the heart. Figure 11-23 is an image of a large vein with its very developed adventitia that you can see, prominent muscular layer, and its intimal layer, and projecting from the intimal layer we can see a portion of valve. So we have the endothelial cells lining both sides of the valve with a small amount of connective tissue being the subendothelial layer of the intima layer. And within this subendothelial layer, we will find an increased amount of elastic fibers within this valve. To complete our review of the circulatory system, in addition to the cardiovascular system we've just discussed, we must also look at the lymphatic system. This is a system that is composed of very thin-walled vessels that's going to be in charge of the interstitial fluid levels in our tissues. When we have excess fluid, the lymphatic system will take this fluid and return it back into our blood vessels. It does this by emptying into the large veins at the junction between the internal jugular vein and the subclavian vein. Within the lymph vessels, the fluid is coined the lymph. The lymph is composed of small proteins and occasional white blood cells. There's typically absolutely no red blood cells present within the lymph. Within our body, the lymphatic system is distributed extensively with the exception of our bone marrow and our central nervous system. Both these regions are devoid of lymphatics. So not only does the lymphatic system play a role in controlling the interstitial fluid levels within our tissues, it also is a major distributor of components of our immune system. The lymphatic system originates within the tissue as lymphatic capillaries. These lymphatic capillaries are lined by thin endothelial cells. In this regard, they are essentially identical to the high capillary venules we've already discussed. What sets them apart from the high capillary venules is that they have endothelial cells that lack tight junctions, and these endothelial cells rest on a very discontinuous basement membrane. This allows for interstitial fluid within the tissues to easily find its way within the lymphatic channels, either between the discontinuous endothelial cells or actually passing through them via transcytosis. Now, some of the endothelial cells that make up the lymphatic capillaries will also lack hemidesmosomal attachments with the underlying basement membrane. This allows for portions of a given endothelial cell to detach itself from the basement membrane and project into the lumen of the lymphatic vessel. This allows this particular endothelial cell to act as a leaflet to facilitate fluid entry and prevent backflow of fluid. In essence, they are virtually like a valve that we saw in the larger veins, but simply a little bit more simplistic form. The lymphatic capillaries will merge together and give rise to lymphatic vessels. The lymphatic vessels will increase in wall thickness compared to the lymphatic capillaries, but they will never actually establish any defined tunicas like our veins and arteries. They will maintain intimal foldings that will form valves within the lymphatic vessels. And figure 11-25b depicted in this slide demonstrates the very thin leaflets that represent the valves within the lymphatic vessels, and they are composed simply of endothelial cells on each side. The lymph will pass through the lymph nodes as it makes its way back to the blood circulation. Figure 11-25a in your textbook is a good comparison between a lymphatic vessel and a venule. You can see in the venule you have a more established tunica media, so at least one or two smooth muscle cells can be appreciated within the wall of the venule. It can be quite difficult to distinguish a lymphatic vessel from a venule. One good rule of thumb is that, as I already mentioned, the lymph that's contained within the lymphatic vessels should not contain any notable red blood cells. Whereas obviously in our venules, we have a ton of red blood cells. So that is a very good feature to look for to help distinguish a lymphatic vessel from a venule. To finish off the lecture, we're going to look a little closer at a specialized connective tissue, that being the blood. The blood is composed of both cells as well as plasma. So the plasma is essentially the extracellular material that is found within the blood. The cells that we may find within blood include our red blood cells, or our erythrocytes, white blood cells, or our leukocytes, and platelets. In order to evaluate various components within our blood, collected blood can have an anticoagulant placed within it, typically heparin, and then it undergoes centrifugation. This process will cause the blood to separate into three distinct entities. First, we have at the bottom of the test tube, so the sediment is composed of erythrocytes. This represents roughly 44% of our blood volume, and this is recorded as the hematocrit. Above this sediment layer, we have a straw-like clear translucent fluid, which is our plasma. This represents a little over half of our blood volume. And then between the plasma and the erythrocytes, we have a very thin gray-white area, and this is called the buffy coat. The buffy coat represents our white blood cells or leukocytes and platelets. This represents about 1% of our blood volume. We certainly all know that blood is essential for our survival. In that, one, it is responsible for delivering oxygen and nutrients to our tissues, and it's also critical for taking away waste products and carbon dioxide. It's critical for delivering signals in the form of hormones from one site to another. It's essential for delivery of our immune system. It's important for heat distribution and regulation of body temperature. And it's also important in our acid-base and osmotic balance in our bodies. Looking a little closer at the plasma, we can see that water is by far its major component. Second to that will be the plasma proteins. In conjunction with the plasma proteins, we will also have some nutrients, some respiratory gases, some nitrogenous waste, hormones, and electrolytes. Through the capillary walls, the lower molecular weight proteins within the plasma maintain an equilibrium with the interstitial fluid of the tissues. Albumin is by far the most abundant and most important protein in helping maintain the osmotic pressure within the blood. So if albumin production were to decrease, fluid within the blood will be drawn out by creation of an osmotic gradient between the blood and the interstitial fluid of the tissues. The importance of proper osmotic pressure in the blood is nicely demonstrated by the cirrhosis patient. These patients suffer from significant liver disease and thus have a decreased amount of albumin production. The decreased albumin causes a shift in the osmotic gradient between the blood and the interstitial fluid, and thus water will move out of the blood into the tissues. And this will give rise to ascites that is seen in cirrhotic patients. So this is when our liver disease patients have a very enlarged stomach full of fluid that's leaching out of the circulatory system. Other plasma proteins will include our globulins. So some of these will be transport proteins, while other of them will be immunoglobulins, part of our immune system. Fibrinogen is another important protein involved in the clotting cascade. And then we'll have some other regulatory proteins, particularly those involved in the complement cascade. Now let's move into some of our blood cells that's found within the blood. So in order to evaluate the cellular component of our blood, we tend to study this via blood smears. And this is just figure 12-3, which is just showing how a blood smear is carried out. The formation of our blood cells is coined hematopoiesis. So here we have just a simplistic diagram that represents the main developmental stages in blood cell formation. This is really meant to highlight the proliferation and progressive differentiation from one stem cell, so our multipotential hematopoietic stem cell, as it gives rise to our mature white blood cells and our red blood cells that we naturally find in the circulating blood. Throughout this lecture, we will revisit this diagram to talk about the individual steps through the various processes of blood cell formation. Let's start first with our erythrocytes, or our red blood cells. Red blood cells are formed within the bone marrow. They are considered terminally differentiated cells in that they will lose their nuclei upon release, and they will not be able to undergo any form of mitotic division. They actually lack a nucleus as well as a majority of organelles. Erythrocytes are packed full of iron that is bound to a particular protein. This is the hemoglobin protein. They're biconcave in architecture, and they have a lifespan of roughly 120 days. Their primary function is in oxygen and carbon dioxide transport. After 120 days, they will be removed from their circulation by macrophages in both the spleen, liver, and bone marrow. When a red blood cell is viewed within a blood smear, it is readily identified as being fairly oval in shape, being quite eosinophilic or pink in color due to the high content of hemoglobin. It will have a pale staining central region, and this is actually a result of the biconcave disc shape of the cell. This biconcave disc actually provides 20 to 30% greater surface area than a typical sphere would for a given volume. So the purpose of this shape is to enhance the surface area for gas exchange and allow red blood cells also to deform and pass through quite small capillaries. Maintaining the biconcave shape of the erythrocyte is a fairly complex cytoskeleton that allows for a lot of flexibility to the red blood cell. The red blood cell will be covered by a standard plasma membrane composed of a lipid bilayer with many globular proteins incorporated within the plasma membrane, and this is where our blood group substances are carried, which allow us to distinguish a type A, B, or O blood type, or AB blood type. Although this is a slightly complicated picture, it does nicely demonstrate the life cycle of the red blood cell. As we discussed in the last few slides, red blood cell production first starts in the bone marrow, and we know that the entire lifespan is roughly 120 days. So if we start first with the end of its life, red blood cells reach the end of this cycle and they're going to be engulfed by macrophages, which are located within either the spleen, the liver, or the bone marrow. So this is demonstrated on the left-hand side of the screen and labeled as step 1. The macrophages will break down the red blood cells into its components. The globin component of hemoglobin will be further processed and degraded into amino acids and released into protein synthesis. Some of the hemoglobin will be degraded to biliverdin and eventually bilirubin and will be secreted into the intestines and be excreted as feces, and it can also be eliminated via the urine. The iron component of the heme will be recycled and transferred to the liver for either storage as ferritin or back to the red bone marrow where it will be again reused in red blood cell production. The clinical importance of really understanding the red blood cell cycle is not necessarily to pass this course, but it's really to appreciate some of the disease processes that can arise associated with our blood. And anemia is one of a very common disorder that to properly understand, you need a good understanding of the circulatory system. So anemia is essentially a reduction in our blood's ability to carry oxygen. There's multiple reasons why we can have this if we think about oxygen carrying of the blood. One, this could occur because we're not producing enough red blood cells, so we may have a decrease in iron. We may have a decrease in vitamins, particularly vitamin B12 and folic acid, which are necessary for simple DNA production. We can also have anemia secondary to blood loss, so we're hemorrhaging. We don't have enough erythrocytes simply because we don't have enough blood. And then we can also see anemia because of increased rates of destruction of the circulating erythrocytes. A good example of this can be sickle cell anemia, where the appropriate nice biconcave shape of the red blood cell is affected. Hence, macrophages, particularly within the spleen, are destroying the red blood cells too quickly. In the next series of slides, we're going to discuss the white blood cells which give rise to both our granulocytes as well as our agranulocytes. All white blood cells share the feature that they begin to function once they leave the blood and enter into the tissues. They are subdivided into two categories, as I've already mentioned, agranulocytes and granulocytes. This is based on essentially the presence of granules within their cytoplasm. The granulocytes include neutrophils, eosinophils, and basophils. They have a very, very short lifespan in our body, and they tend to have nuclei that are lobulated, and they will contain specific granules within their cytoplasm. Comparatively, the agranulocytes, this includes our lymphocytes as well as our monocytes. So the monocytes are cells that are going to give rise to macrophages. These cells lack specific granules. They do contain some granules, but you're not going to appreciate them on microscopic H&E slides. And their nuclei are spherical, although they can be slightly indented, but they are not lobulated. Globally, white blood cells function to defend our bodies against microorganisms, and they also play a critical role in helping repair our injured tissues. Now, table 12-2 in your text is a very nice summary table for the various types of white blood cells and their function. Let's begin first with the neutrophils. So again, the neutrophils are a type of granulocyte. In the upper right-hand side of the screen is the appearance of the neutrophil on a blood smear. The appearance, this photo nicely demonstrates the lobed appearance of the nucleus, so usually about 3 to 5 lobes. And you can see a faint granular texture to the cytoplasm. Sometimes on H&E, on high power, sometimes it's difficult to appreciate the very fine granules that are present within neutrophils. Neutrophils represent the greatest number of white blood cells within our circulation, and this is over 50% of our white blood cells. Neutrophils are the first leukocyte at the site of infection, and they play a role for destroying bacteria by phagocytosis. They also will release mediators to propagate the inflammatory process. They are very short-lived. They were present within the blood for roughly 6 to 8 hours, and once released into the tissues, they have a life cycle of about 1 to 4 days. An electron microscopic view of the neutrophil demonstrates very nicely the multi-lobed architecture of the single nucleus. And you can see all of the cytoplasmic granules that contain various enzymes slated for antimicrobial activity. Now, the one thing to appreciate in this picture is that neutrophils really lack a lot of cytoplasmic organelles. So they're not able to produce new proteins, very limited protein production. Thus, neutrophils are incapable of continued function. Thus, when one neutrophil is carrying out a burst of activity, that is then the end of its life. Although I won't test you on this figure, but this is figure 12-6 from your textbook, and it demonstrates the importance of endothelial cells within the postcapillary venules in the recruitment of neutrophils. Here we can see cytokines being released from activated macrophages within a tissue, in particular interleukin-1 and tumor necrosis factor, which I'm sure you've heard about in your biochemistry courses. These are the two major players in acute inflammation. They encourage the endothelial cells to then express surface proteins, which will bind to passing neutrophils. The neutrophils will then run along the endothelial cells and begin to squeeze through the endothelial cells, which are actually changing their architecture and producing some space so that the neutrophils can actually pass into the tissue. This is a process via diapedesis. And then once into the tissues, they're able to undergo chemotaxis through the tissues, meaning travel through the tissues quite effectively to the site of infection or inflammation. Moving now to another granulocyte, the eosinophil. The eosinophil represents about 1 to 6% of circulating leukocytes, so this is substantially fewer than neutrophils. It's produced in the bone marrow, and it tends to reside within the spleen, lymph nodes, and along the gastrointestinal tract. Its major function is in inflammatory responses due to allergy, and as well as inflammatory responses against parasites. So eosinophils are actually able to bind to parasites and release the granules within their cytoplasms that will initiate disruption of the parasitic membranes and death of the parasite. Eosinophils also have a multi-lobed nucleus, but it's only bilobed. And they have a cytoplasm filled with very eosinophilic, almost ruby red granules. So much larger and brighter granules than the neutrophil. The eosinophil is also slightly larger than the neutrophil. Here we have the electron microscopic view of the eosinophil with its bilobed nucleus and the cytoplasm containing not only large granules but also an extensive network of smooth endoplasmic reticulum as well as ribosomes. So much more cellular organelles and more activity within the eosinophil than neutrophils. A comparison of an eosinophil to a neutrophil in either of these images, you can appreciate the marked difference in the types of granules that make up the cytoplasm. Sometimes the nuclei are a bit difficult to distinguish within the neutrophil in comparison to the eosinophil. But typically, the eosinophil nucleus is simply bilobed, while the neutrophil nucleus is multi-lobed. When we look into tissue sections, so H&E slides of various tissues, you can see that the appearance of the neutrophils and the eosinophils are quite different from their blood smears. So the cells are quite small in appearance. You can barely appreciate the cytoplasm. But on the left-hand side of the screen, you can see that this whole area is teeming with very small neutrophils with tiny, almost looks like multiple tiny nuclei, but these are the multi-lobed nucleus that we're seeing in the sections. And then on the right-hand side of the screen, the eosinophils really pop out because of the intense staining we see in the granules that make up the eosinophils. And lastly, we have the basophil. The basophil is the least common circulating white blood cell, represents less than 1% of the circulation. Its granules are quite comparable to the mast cell. Its size is somewhere between a neutrophil and an eosinophil, And the true purpose of basophil is not quite known, but it's suspected that it supplements the function of mast cells in type 1 hypersensitivity reactions. Basophils will have an irregular bilobed nucleus, and its cytoplasm will be full of quite large, densely basophilic, so deep blue-purple granules. It is very unlikely as we look at blood smears in our laboratory component that we will find a basophil. Here we have an electron microscopic view of the basophil with its bilobed nucleus and large granules that contain histamine, comparable to mast cells, proteoglycans, even heparin, as well as chondroitin sulfate. Its membrane has a high affinity for IgE immunoglobulins, again comparable to mast cells. Let's move now and take a closer look at our agranulocytes that make up our white blood cells. Let's start first with the monocyte. This is our largest white blood cell, and it constitutes up to 10% of circulating white blood cells. It's present within the peripheral blood for about 3 to 4 days prior to moving into the tissues. Once in the tissues, the monocyte will differentiate into macrophages, and these macrophages have, of course, the capacity for phagocytosis. Monocytes, once they take on the role of a macrophage, can also perform the duty of an antigen-presenting cell in order to activate our cell-mediated immune reactions. As with all the agranulocytes, the monocyte has one nucleus, usually eccentrically placed, and is often indented or kidney-shaped. And notice its large size in comparison to the red blood cells in this blood smear. Here, the electron microscopic image of the monocyte demonstrates its eccentrically placed indented nucleus, and very few granules noted within its cytoplasm, and various organelles are certainly present. There's a lot of pseudopodia existing on the surface of the monocyte, and this is because these are quite motile cells once in the tissue. Now, the monocyte again gives rise to macrophages, but depending on the tissue type, this final differentiated cell may have a different name. So when it's in simply connective tissue, we call them macrophages. If this occurs within the liver, they are the Kupffer cells. In the skin, they're termed Langerhans cells. Within the lung, they're called alveolar macrophages. In the brain, we call them microglia. In the bone, they're termed osteoclasts. And then lastly, in lymphoid organs, they are also present, and they're simply termed macrophages in these areas as well. The other type of cell that makes up our agranulocyte category are the lymphocytes and natural killer cells, as well as the plasma cell. So these particular agranulocytes are derived from a slightly different branch of development than the monocytes. The lymphocyte formation goes through 2 precursor stages. So they first are a lymphoblast and then move into a prolymphocyte before they eventually become recognizable as a particular type of lymphocyte. The main feature of lymphopoiesis is the progressive diminution in cell size. So unlike other blood cell types, lymphocytes also proliferate outside the bone marrow. This occurs in the tissues of the immune system in response to specific immunological stimulation. The lymphocyte, whether it's B or T lymphocyte, is the smallest and second most common circulating white blood cell in the blood. So it represents about 20 to 50% of circulation. It circulates between various lymphoid tissues as well as other organ tissues via the blood and the lymphatic system. Its role is in the immunological defense system and immune surveillance. In a blood smear, lymphocytes are characterized by round, densely stained nuclei with a very small amount of non-granular basophilic cytoplasm. The smaller the lymphocyte, the more inactive it is. In the peripheral blood, B and T cells, or natural killer cells, all have the same appearance as they're en route towards the tissues. Electron microscopic images will demonstrate the large nucleus of the lymphocyte with very small amount of cytoplasm with very scant number of organelles and only a few ribosomes. The two main subtypes of lymphocytes include T cells, or cytotoxic T cells, as well as B cells. Here is a tissue section of epithelium covering connective tissue. And within the connective tissue, we see this dense population of blue dots. These blue dots represent the infiltration of lymphocytes into the connective tissue. So at this power, you can't appreciate any kind of cytoplasm, and even on higher power, very minimal amount of cytoplasm will be appreciated around the nuclei of these lymphocytes. Again, we can't distinguish a B or T lymphocytes, or natural killer cell for that matter, from one another simply based on microscopic appearance. We would need special stains in order to tell them apart one from the other. And the final cell to discuss in this diagram is the thrombocytes. These are our platelets. Platelets are small, membrane-bound, but anucleate cytoplasmic fragments that are derived from megakaryocytes. They have a round, oval, and biconvex shape, and they're quite small. They function in clot formation and repair of minor tears or leaks within blood vessels. We can see in the image of the blood smear here the relative size of the very small cellular fragments that represent the platelets in comparison to the red blood cells. So again, no obvious nucleus, and the electron microscopic image demonstrates the presence of some granules within its cytoplasm, but given their small size, we can't really appreciate any kind of granularity when we're viewing them microscopically. I won't be testing you on this diagram, but it is, I think, an important diagram to understand as you make your way through dental school, in that platelets play a major critical role in controlling hemorrhage and wound healing. They are involved in primary aggregation, where they aggregate along damaged endothelium. Once bound, they undergo a transformational change, which allows them to bind to fibrinogen, so a plasma protein we talked about earlier. Fibrinogen will break down into fibrin, which will seal and maintain a clot. So platelets play a very important critical role in the clotting cascade. And it's important, I feel, to understand this process in quite a lot of detail, since as a dentist, you're going to cause bleeding every single days of your career. Here's an excellent scanning electron microscopic image of platelets as they aggregate and join together via fibrin bridges. I love this image. There are a lot of different clinical considerations that you're going to be learning about around platelets. People can have too few platelets, so thrombocytopenia, which will tie into bleeding disorders and bleeding problems closely linked with dentistry. And then you're also going to come across diseases where people have thrombocytosis, so they essentially have excess platelet deposition leading to various problems with the cardiovascular system. I will leave you with this summary sheet of the various types of blood cells that we've talked about today. I think it's a good summary. I'm not going to ask you to memorize these numerical figures whatsoever, but I think it is important to understand the lifespan of these cells, certainly the microscopic appearance, both in tissues as well as blood smears, and their primary function. Thank you very much.